everybody. Reporting to you again from the Glamour City, Hollywood.
Senescent cells are named because they appear gradually in aging, and they accumulate in certain tissues. And in disease states, this is more acute. And there are many diseases associated with the accumulation of senescent cells. The question also is, when the senescent cells accumulate, as to why they accumulate. And when they accumulate, these cells have a lot of what's called non-cell autonomous properties, where they affect the local microenvironment and functioning of the tissue surrounding it. So, for example, when they accumulate in the adipose tissue, they affect the functioning of the adipose in its metabolic control of the body, and you end up with type 2 diabetes as one of the outcomes of senescent cell accumulation.
If you love listening to this show, please consider giving a rating and a review on Amazon Alexa or wherever you listen. We want to continue bringing you this amazing content, and part of our ability to do that means that we need a big audience. Now, it may not seem like much, but rating and reviewing the show will help more people find us, just like how you found this show. Simply on any podcast platform, search for a show, scroll down to the bottom, and push 5 stars. It's that easy. Thanks for supporting the show. Today, I'm joined by CEO Robin Mansukhani and Chief Scientist Dr. Anil Bhushan of Deciduous Therapeutics. Welcome to the show today.
Thank you for having us.
So let's start with a big picture understanding of why we age. First, what are senescent cells, and how do those cells affect aging, autoimmunity, and metabolic diseases?
Senescent cells are named because they appear gradually in aging, and they accumulate in certain tissues. And in disease states, this is more acute. And there are many diseases associated with the accumulation of senescent cells. The question also is, when the senescent cells accumulate, as to why they accumulate. And when they accumulate, these cells have a lot of what's called non-cell autonomous properties, where they affect the local microenvironment and functioning of the tissue surrounding it. So, for example, when they accumulate in the adipose tissue, they affect the functioning of the adipose in its metabolic control of the body, and you end up with type 2 diabetes as one of the outcomes of senescent cell accumulation. So,
and just for clarity, is that senescent cells in themselves are not necessarily bad, right? So it's fairly natural for them to accumulate. It's just that some of them over time may not necessarily have good use, but really the issues around inflammation and some of the other aspects you were referring to, correct?
Right. So evolutionary senescent cells are they're useful in the senses that they are created, but they have a sort of a timer attached to them for their own demise. And the problem really becomes is when they're not removed in the proper time. So they are very useful for example, in wound healing, where they recruit the immune system to come and do the repair. And when you do the repair, they also have this system where they are cleared themselves, and then the inflammation goes away. And this problem, which is the basis of deciduous therapeutics, comes because there's a failure of the immune system to remove senescent cells. And what deciduous does is to reactivate the immune system that's failing to remove these senescent cells, so that the clearance can go on and as it should be going on.
So for listeners that are not as familiar with this space, it's kind of like spring cleaning. It's good to timely clean things out and allow for healthy environment. It's just that when the immune system fails to clear these out, it starts to accumulate and have adverse effects. Now, how does that tie into specifically aging? Because when we think about aging, we think about the specific diseases, but here you're tackling something that's kind of indirect, right?
Yeah. So aging is more a gradual loss of the immune surveillance that removes these cells. And so you would, the analogy you make of spring cleaning it's like getting a tune-up for the car in terms of getting oil changes to keep the car functioning at longer time. So that's what deciduous will do, is to tune up your immune system so that the senescent cells are constantly being taken care of.
Okay.
There's also one
Please go ahead.
addition I want to make here, and that's the context of senescence has often been thought about in the context only of aging, and people at advanced stage will have more senescence than others. In reality, though, it's actually driven more by biological age than statistical age. So what I mean by that is you can actually see senescent cells in younger people who have certain diseases. So even in type 1 diabetic patients, diabetes, cardiovascular patients, IPF patients who aren't always the oldest patients, especially in the case of T1D, you actually see senescence there. So it's actually not a statistical age, it's more about biological age, where the immune function is not happening properly. And so our goal is to bring that back online.
Well, this is fascinating. And I think there's a couple of related questions that I'm going to ask regarding both impact of cancer, but also things like COVID and other things that's compromising your immune system. But let's go back to the genesis. Anil, I believe some of this underlying IP or research work came out of your lab, your work, and some of the work since then has been supported by grants from NIH, UCSF Diabetes Center funds, and eventually it's evolved into commercialization through now what is Deciduous Therapeutics. So I'll leave it to both of you to explain kind of how it emerged into this current venture.
Yeah. So Anil had been doing diabetes work for most of his career, and I think been following kind of a more traditional path for some time. And one of the things that really caught my attention when I first met him before starting Deciduous is that he was looking at, had discovered that senescence existed in not just age-related diseases. And so our first meeting was around actually seeing this in the context of type 1 diabetes. And I found that fascinating because I'd done a company, 15 plus years ago around type 1 diabetes treatment using a glucose responsive insulin. But I'd never thought about type 1 diabetes in the context of senescence. And so that was very interesting. Mm-hmm. And so as we got into discussion, what we learned is that, from his work that, if you actually take senescent cells out from different tissues and you do single cell RNA sequencing work on them, you can actually understand how they communicate with the immune system. And so it became not just a diabetes story, but it became a larger story around senescence in multiple different disease classes. And so we identified that as being a very intriguing platform. We were able to form the company based on that initial idea.
Super.
So there's a lot of interest in once it was recognized that when senescent cells are not properly removed, that you would, people would want to do the job that the immune system is failing to do, and the classical way to do that is senolytics, which are drugs that target the senescent cells directly. And we thought that a much better approach would be to understand what the immune system normally does and why it's failing, and to target the immune system. And
just to continue on what Anil was saying, was really rather than focusing on the senescent cells themselves, so you're focusing on the immune system. And there, I believe the core IP is around the development of novel molecules that activate a critical endogenous immune surveillance mechanism to then selectively remove these senescent cells. Can you go into a little bit more detail?
Yeah, so let me just back up quickly. So like I said, the ideas around targeting the immune cells, put very simply, there's a way that nature intended for this process to happen. And so our initial work was identifying that process, and that turned out to be the natural killer T cell. And so the company Deciduous is then focused now on making small molecules that can selectively activate these targets. And the data that was published last year is really quite compelling, where we show that if we do a single dose using an NKT agonist, we can not only remove senescent cells in a period of about 4 to 5 days in different model types, but that actually, the reason that actually matters is because you can reach, registrable important disease endpoints about 7 to 10 days after that first treatment. And it's only a single treatment we're giving. It's just much more of a vaccine regimen than a typical small molecule regimen. We give, one dose on day 0 of the study. And by day 10, we see that single dose on day 0 had an impact on endpoints that would be attractive to reach in the clinic. And interestingly, beyond that, what happens is that effect on that endpoint actually lasts for over a month. So you're talking about removing senescent cells with a single dose. The effect is seen after 10 days, but that effect lasts for over 30 to 40 days. So you would repeat this treatment very infrequently, every couple months or so, which is very different treatment regimen than current standards of care across metabolic and fibrotic diseases.
That's really interesting. And based on your study, what accounts for why there's that residual benefit that lasts so long?
It has to do with a couple things. One is the potency of the target. So, when you hit these NKT cells properly, they become very active and proliferating for a short burst of time. So it's basically like you start the party, things happen very quickly with removing senescent cells. Then the NKT cells then go back to a normal resting state. But because of the potency of how quickly and how much of the senescent cell volume they got rid of, those senescent cells don't repopulate then for over 6 weeks at a time. There's a natural time course that it takes for a cell to become damaged and irreversibly damaged and permanent cell cycle arrest to actually becoming senescent, and that is thought to be well over a month. So if you can hit them with one big swing, they will typically not repopulate for some time, and that's seen across different approaches, not just ours, but it really has to do with the potency of the drug and the mechanism you're using, and this is one approach that is really properly exploited.
And then, are there any side effects of that level of potency? And then, really the related question is going to be, have you experimented with lower dosage and maybe more frequent therapeutics, for instance?
Yeah, we haven't seen anything with side effects that are concerning or anything in the toxicology that would be a red flag. You do see that obviously the NKT cells go from a very base resting level of a very suppressed number to then a very active and proliferating state in about 4 days. But what's nice is the cytokines are minimal and quite brief. As well, the NKT cell population goes from the low state to the active state to the low state again within about 8 days. So it's a rather quick turnaround. It's kind of like you hit it once and it goes back to sleep once it's done its job.
Now, going back to diabetes type 1 and even diabetes type 2 as an example, when this therapy is, let's say, coupled with, let's say, lifestyle changes, would the NKT cells, for example, stay more vibrant and active longer?
That's an unknown in my opinion. Anil, do you want to take a crack at this?
Yeah, that's a good question. We don't know so one of the things that the NKT cells are locally reduced in the adipose tissue. So it's really hard to address this question in people is, whether the iNKT cell function is rejuvenated with, lifestyle changes, because you have to actually measure it in the adipose tissue. But because we're not looking at, blood samples, it's not the circulating iNKT cells that's failing, but it's the resident iNKT cells within a tissue. So it's a good question. Now, I don't know how to address that, though.
Now, one of the things I think, again, for listeners, including myself, is that are there differences in senescent cells? So in other words, when the activation of the iNKT cells are there, is it having more effect in elimination of certain senescent cells in vivo versus others?
Yes. So the way the iNKT cells recognize the senescent cells is by a particular flag that the senescent cells put up, and depending on how the senescent cells are generated, in different cases the flag is different. So the iNKT cells would not necessarily remove all senescent cells, but only the cells would put up a flag that really flag the iNKT cells. For example, a lot of stromal senescent cells in cancer don't have the same, what we call the oncogene-induced senescence doesn't have the same flag. So it won't be removing those cells, for instance.
So my understanding is that the current focus is on class of lipid-activated T cells to reactivate the iNKTs. And I believe it's specifically focusing on IPF, which is lung disease related, and then kidney, as there are just better diagnostics to measure the effectiveness.
So as far as indication preference for us,
yes.
Yeah, so we've taken a little bit of different approach to this. We wanted to show the robustness of the platform. So the first POC was done, proof of concept was done in type 2 diabetes, which is a more longer term chronic type of model to test in. The second one was done in lung fibrosis, as you mentioned. That's a very acute model. You do a single or second treatment of bleomycin to induce fibrosis in the lung and replicate pulmonary fibrosis. We are now moving into other metabolic conditions like kidney and heart, and others coming forward. But part of the rationale in our platform approach is to show it doesn't seem to matter how these senescence is induced. It can be chronic from a diet. It can be from an injury like bleomycin. It can be from certain treatments that are given only a couple times that kind of thread the needle between metabolic and fibrotic. But the effect looks to be the same. The drugs work very similarly. The dose is the same, and the endpoints are also met, in all these cases within 7 to 10 days. And so there's a common funnel here where it comes down to that pathogenic senescent cells are driving these diseases. And if you can get rid of them, you will not only ablate disease, but you may have the chance to restore normal function.
So there's another important point to make, which is that, most diseases, for instance, type 2 diabetes involves more than just, one organ. And it has, a lot of effects on, say, cardiovascular issues, arteriosclerosis, kidney function. And a lot of this is associated with senescence in these tissues at the same time. So activating iNKT cells systemically will remove senescent cells throughout. So you're treating the disease, in entirety rather than just focused on the symptomatic treatment due to one organ.
To have this therapy that can attack all of these functions and diseases. And like you said, diabetes is a great example that affects so many that are critical to our health. So how would this potentially work in conjunction with traditional pharmaceutical therapeutics?
Yeah, it's a good question. And it likely depends on which other indications are, what the standard of care is in other indications. We see this as being potentially a standalone. Given how we select indications, we're looking at things where we can, we really attack indications. We have a 15-step filter as to why we pick certain indications. But we really look to work in indications where we can change the paradigm for standard of care. So meaning, in the case of lung fibrosis, right now you are taking 3 to 6 pills a day. They have minimal effects on the disease endpoints. They have bad side effects. The data we have suggests that this approach will be much more effective, even given intermittently every 6 to 8 weeks, rather than the current standard of care, which is every multiple times a day. So we aren't really looking to add on necessarily to another existing therapy or another existing treatment regimen. We're looking to replace existing regimens. Now that'll be a little bit different indication by indication. That might be more stepwise as we go to the clinic and as more data comes out. But overall, the data we have in the places where we focus areas where we feel like this can become the standard of care.
So from an innovation, I mean, that's outstanding. And certainly from a patient perspective, it's something that we can only dream of. But at the same time, it poses a big threat to the big pharmas because, those are blockbuster drugs that they've invested heavily billions into, and potentially you could, replace it overnight. How do you actually address that from a go-to-market strategy and making sure that you work in cooperation with the pharmas rather than in competition?
Yeah, I have to keep, I guess, some of this relatively confidential. We have open channels right now with some of the big pharmas who are in the areas of interest that we are focused on. So those conversations have already been initiated and are progressing. So, we have a nice platform where we can clear senescent cells across multiple different disease classes. There are certain pharma partners that are really active in each of those spaces, and we have those discussions initiated already to understand what a partnership would look like and when the right time to actually make a formal partnership actually happen with those companies.
A type 2 diabetes clinical trial is outrageously expensive, and I think the only way to do it is with partnership.
Yeah, absolutely, without a doubt. Now, I kind of alluded to it at the beginning, which is around cancer as well as COVID. So when you're going through, let's say, cancer treatment, certainly immune system is compromised. Certainly COVID has a lot to do with our immune system and inflammation as well. In those very different kind of situations, how would your therapy work or help strengthen the overall immune system?
Yeah, I mean, I can speculate because we haven't really worked with SARS-CoV-2 directly, but clearly the impact of SARS-CoV-2 is much more common in people with type 2 diabetes and obesity. So there is some, obviously the connection is there. The question is, what is the connection? Is it a direct connection? And this all I can speculate is that in other viral cases, the ability to infect senescent cells is much greater. So you actually get a greater chance of replication and infection because you have more senescent cells. So clearing senescent cells would, lead to less sort of a repository for viral virus to hang around longer than it should.
Great point. And again, I recognize that you do have to speculate because you guys haven't gone into the research aspects. But you mentioned I think the cell-based research. There's other research that's also been published as well. Are there any other research that's coming up that you can actually allude to?
For Deciduous, the company?
Yes.
Nothing I can say publicly. So.
Okay, understood. Now, I believe you guys are in preclinical data. Is that correct? And then there is a planned clinical phase 1 sometime in 2024. Is that accurate?
Yeah, that's roughly accurate. We have a nice class of lead compounds right now. We're working on selecting between which lead should be the ultimate development candidate. And so once we have that selected, we will move into the more formal, toxicity and IND enabling studies, and then file the IND once that is complete.
Now, in terms of efficacy and effectiveness or performance, what are some of the measurements that's used to determine how effective something is, and if it's enough to actually replace an existing pharma-based therapy, for instance?
Yeah, this is a very good question. So, the premise of the company obviously is around, can you naturally move senescent cells via the immune system? And I think that's fascinating to talk about. I think it's academically very intriguing, but it really doesn't matter unless you can actually reach disease endpoints that are measurable and widely accepted as being, the ones to actually impact. So when you think about our efficacy, it's not about removing senescent cells necessarily. That's the middle person there for the process, but really where the rubber meets the road for us is, does it actually impact quantitatively, irrefutably, disease endpoints across multiple different disease classes? So that is what we're looking for in the clinical studies, to show that. How does it replace current standards of care? It likely will. There's going to be some movement there depending on what else is coming down the pike with other companies. But I'll just say that I think we feel very strongly that if our preclinical data holds up, that single treatments that can reach endpoints and have them be durable, meaning that they can last over a month, will be positioned very well regardless of what else is out there.
And then just one more question for Neil is, the sensitivity around some of these compounds, how complex is it, and how minute in terms of small changes could potentially have a huge impact on exactly the kind of metrics that Robin was just referring to?
Meaning small changes?
Formulary or the methodology, the core.
Yeah, I mean, and a lot of it has to be fine-tuned, which is what Deciduous has been doing, is not only making small changes on the molecules affects the function in depending on where you're making the changes, has great effect on the function of activating the iNKT cells, but also how you deliver it in terms of the formulation is greatly important as well. So all of those things create, is what we can create the best, sort of drug that we can use clinically.
And then in that process, is there use of let's call it machine learning that can simulate some of these things before it actually goes into preclinical or even clinical phase? And
it can be useful. It can kind of, I look at it as, if you have 100 candidates, then that might be able to get you to the top 20, but nothing replicates actually going in vivo and seeing results for yourself. What you model does not take into account, very complex biological systems. So I find that stuff useful and interesting, but only as a part of the equation, not the entire equation. Yeah. So we do employ some of these practices when needed, but there's no substitute for actually seeing real data from in vivo studies.
So in silico modeling and modeling was very important when we were designing the molecules, and that involved a lot of heavy computational approaches. Mm-hmm. But we're past that at this point.
Understood. Now, in terms of risk, what are challenges created by clinical trial failures of other senolytics?
Yeah, so it's been a bit of a mixed bag. I'd say the couple years ago, Unity had the OA study, so it did not proceed into phase 2. So that was not good for the entire senescence ablation field. More recently, I think their BCL2 study looks more promising, and the first readouts that we've seen so far that they presented. So the headwinds are always going to be there. I would think of it a little bit differently for us. It's not our job to worry so much about what other companies are doing, what data they're getting. Our focus is on what we are doing, our approach. We think we have a very unique approach that's nicely positioned against the more traditional senolytic approaches because we actually do it the way nature intended. So I would actually answer the question by saying it almost doesn't matter to us, if other work out there is positive or negative. I think both scenarios can have headwinds for us, some good, some bad. So it's really incumbent upon us to just get our stuff into the clinic, prove that the in vivo data can hold up in people. I think that's really where our focus is.
And then with the current macroeconomics and the geopolitical turmoil that we're going through, that's affecting public and private markets, how is this affecting your business and the future outlook for the next 5 years?
Yeah, again, I'm not sure that it affects us so much in the near term. I know that the headwinds are out there. I know that the venture markets aren't what they were last year or the year before. But again, we're well funded to do the work we were, raise money to do. So there's no internal thought process right now about this is the markets are affecting us internally right now. I think it's something out there that we need to be cognizant of for the next fundraise, certainly. But at the current time, we have the funding to go forward and make good progress. And at the same time, we cannot control the markets as a small company, and we're not going to try to. So our job all along is to get the best data possible, show that it's valuable, and then use that to raise the next set of financing.
Super. My final question for both of you, and I'll give both of you the opportunity to respond, is lessons learned, whether it's from your academic background or from past or current commercialization, lessons learned in terms of product or project failures and insights that you can share with others.
I think the timeline for go, no go is a lot more stronger in the startup world, and coming from academia, that was a lesson I had to learn, is to make quick decisions and be if something is not panning out, you better stop it and move somewhere else faster. So I take that lesson back to the lab as well in academia and take that approach, which works a lot better too, which though we have be cognizant in academia of different missions. So that was the big lesson I learned.
My big lesson learned is, I think despite what it might seem like at times, I think most of the pharma biotech world is focused on evolutionary changes because it's something that they're used to. They understand how certain models work. They understand how certain diseases are supposed to be treated. And I think when you come with something that's not evolutionary but more revolutionary, it's going to intrigue a lot of people. It's also going to have half of those people are going to say this will never work and that'll be crazy. But I think that's the way you change diseases, not by evolutionary changes, but by revolutions. And so just know that it's going to be controversial, and that's okay. And at the end of the day, the data that's quantitative and irrefutable will tell the real story versus anything else.
We look for revolutionary, and I think that's a key. So, and those are the kind of guests that we have on our show as well. So with that, I have been joined by Robin Mansukhani and Dr. Anil Bhushan of Deciduous Therapeutics. Thank you for joining today.
Thank you.
If you've enjoyed this episode, take a moment to rate our show on any podcast platform that you listen to. Scroll down to the bottom and push 5 stars. It's that easy. And as always, thanks for listening.